Our first speaker is Dr. Lucie Bruijn, and she is the ALS Association's chief scientist. She was born and educated in South Africa, which is just this melodious accent that we're privileged to hear, and you always pay attention when she's speaking. She received her bachelor's degree in pharmacy, but alas, Walgreens and CVS was not to have her as an employee for more than 1 year when she found out that this was not her calling. So she left the pharmacy field, moved to London, and she there received her master's degree in neuroscience and a PhD in biochemistry. She did her postdoc work with Don Cleveland, first at Johns Hopkins and then at the University of California in San Diego. She developed one of the first models of SOD1, mouse model, in 1994. From academia, she moved to industry and Bristol-Myers Squibb. And then, in 2001, she arrived at ALSA, and we will never let her go. She has been a jewel at the ALS Association. Because of her, we have the respect and the participation of extraordinary scientists worldwide. Here are just a few of Dr. Bruijn's accomplishments. She is currently chair of the Integration Panel of the Department of Defense's Research Program. She is on the Scientific Advisory Board for Prize for Life and the Italian ALS Association. She is an advisor to several companies, including Bristol-Myers Squibb. She's an advisor to the biorepository for the ALS Registry at the Centers for Disease Control. She's on the Data Safety Monitoring Board for NeuralStem. And she has established a genome-wide association consortium between the UK, Ireland, Netherlands, Italy, and the United States. And she's initiated the Gene Identification Consortium. And she's helped to develop the IPS, which are the skin cell, I'm consolidating that, I know you'll explain it further, consortium in partnership with NIH, ALS, Parkinson's, and Huntington's disease. Now, I didn't want to take up all of her time with her introduction, so there is more to come. Please welcome Dr. Lucie.
Thank you, Ellen, for that generous introduction, and it's really a pleasure to be here. I just thought I would highlight, it's always hard to measure success because actually what you prefer to hear from me is that there is a treatment today that will make all the difference for every one of you. And so, of course, that is our ultimate goal, to find treatments and a cure. But I think it's reasonable to look at the successes along the way because that both encourages us as scientists, as funding organizations, and also tells us in what direction we have to move forward faster. I think the discovery of new genes has been phenomenal, and I will highlight that as we move forward in the talk about how quickly in the recent years we have found so many new genes and why they are important. We have unique partnerships globally, which has helped us find some of the challenging genes, and the big finding in 2011 of the chromosome 9 gene that cannot happen with one small group alone, and it really happens through large consortia. We have been really fundamental in building resources for the ALS community so that scientists can actually do the research. This comes sometimes in the form of mouse models, tools and technologies, and also now the IPS cell lines. We were the first organization in the ALS field to embrace translational research. I will explain what that is about. There are many now and many organizations and many diseases that are focusing on this. But that really was an initial change for the direction of associations to think more about how we can take good academic side from bench to bedside. The field was first for finding antisense technologies to treat specifically against SOD1. Very exciting. Often people ask me, well, why is it that we're so behind? Alzheimer's has so much investment, is much further. Well, actually, in many cases, we've been a first for some very novel discoveries that have paved the way for other diseases such as Huntington's, SMA, and muscular dystrophy. So there's a lot of hope in the advances we've made in technologies. We were also the first to publish in induced pluripotent stem cells in a particular disease. Again, really exciting work from teams at Harvard and worldwide. And we also supported the development of the surgical technology required for the stem cell trial, which I'll talk about at NeuralStem. Often it's small things that pave the way to make all the difference. Without that technology, it would not be possible to have injected the cells into the spinal cord. And I think what is very exciting through a wonderful partnership with Niels and a lot of the work you'll hear about in Dr. Berry's presentation is our partnership with Niels to really help build that infrastructure and improve the number of clinical trials that we can do in an effective way using many, many centers throughout the US. And really, I can't put a slide together with all the scientists worldwide that we fund, but I'm very proud of our initiative and the Milton Safinovitz Fellowship Program, which seeds the young scientists for the future. And these are our research scientists. We also have a clinical fellowship program, and you'll hear mention of that in the next presentation's introduction. But it's not all about just doing it on our own, and we do have significant funding partners. For example, the NIH. We work with the Packard Center at Johns Hopkins for partnering of programs. The Therapy Alliance, the CVS funds that come when some of you might have seen it when you go to CVS, they request money that goes to an organization, the Therapy Alliance, and we partner with them to benefit from that and make those dollars go further with ours. We work with the M&D Association in the UK on big projects. We have for at least 12 years been co-funding a database for genetics. We're now working with the frontal temporal dementia organizations because we know more about ALS and its connection with FTD. And in fact, as well, because of that, I've recently been engaging with the Alzheimer's Association in the UK as well. And more recently, we are funding with the MDA a diaphragm pacing study. So it is really all about partnerships at funding level through scientists worldwide that we will make a difference in the disease. And many of you have probably seen this image, but why I like to highlight it is that really it's a process to get drugs or ideas from the beginning stages and early discovery all the way into the clinic. And because of that, we have multiple different programs, different strategies. All of them rely heavily on building our tools and technologies. But central to it is those of you living with the disease. And we've learned more and more that we cannot do these studies in isolation and have the basic scientists working here, the clinicians working there, and then connecting everything. And important to our studies, both in the lab and in the clinic, is patient material, the samples, the skin cells, the blood, the CSF fluid, the imaging, and also understanding from clinical trials what goes wrong, how we can improve them. So it really is a team effort. And this is just to highlight, it's a lot of detail really, but the important thing is that we have many different types of programs. So much of your dollar support goes into a whole variety, from the investigator-driven program, which is NIH style, where we wait to get good ideas to come to us, to more ELSA-driven programs where we identify the opportunities and the gaps, our clinical fellows, our research fellows, and then importantly, our drug discovery contracts, which help take the academics and the industry and bring them together in partnership. The key areas we focus in, well, genetics has really been a fundamental area to give us clues. Once you have the genetics, we have to build models. So building models either in a petri dish or a mouse model or a rat model, zebrafish and flies. Biomarkers, the signatures for disease, and I'll talk a little bit about that. Stem cells, I know many have real interest in this, and I'll talk about that. You'll hear a lot more in the next presentation about neuroinflammation. Also, knowing more about the biology of how these motor neurons work and connect with the muscle and the axonal dynamics, how things traffic from one cell to the other, is becoming incredibly important and of interest. And then RNA biology, a whole new area that we never really realized we would be tapping into so much for ALS. So today I'll talk a little bit about the biology in the limited time we have, just so that you get a visual again. Many of you have seen this. I want to touch on the concept of how drug discovery happens, and again you'll hear in our next presentation a shift more onto the focus of clinical trials as part of that process, and then a few highlights from the program. So this is just to again emphasize the point that this is a very complex disease. It's the cells in the brain, the cells in the spinal cord, that all have to exquisitely connect by these long projections and then function by a long projection to the muscles. So it's not one region in the brain or in the spinal cord where the disease is core, it's really this whole connection and this network. And this is particularly important when we talk about stem cell therapies. Where exactly are those stem cells going? What are they going to be doing? What do we think we would like to have happening? And are they really making things reconnect? Some of it is a little bit misrepresented sometimes. When I joined the field in 1993, the focus was all about the motor neuron. In fact, I started my research in Don's lab looking at what these things might be in the cell, and in fact they're the neurofilaments. They're these very important structural elements that form part of the cell. And we were almost sure that there must be mutations in ALS patients in these neurofilaments that cause the problem. Well, we spent quite a lot of time and energy on that and didn't find that, but I will bring neurofilaments back into the conversation later on and tell you how they could be interesting. So what is another important point to note, that really through building the mouse models, which we could later do with the gene discoveries, we became much less motor neuron centric. Absolutely the motor neuron is compromised and dies, and that's why your muscles can no longer contract because the connections are broken. But we care a lot more about the surroundings. These are the astrocytes that sit around the motor neuron. This is a motor neuron, and I sort of promised you it had one long axon. Well, that too is a little bit of a simplification. It does have one long axon. Actually, I learned from some biologists that maybe we have more than one axon, but we won't go down that route. But the reality is we have lots of branching. And so, in fact, it's not as simple as rebuilding one connection. It's rebuilding a whole network. We also have to understand more about this junction. That what you can't see here is a muscle, a dark area, and what you can see is the connection that is made by this nerve cell with the muscle. And that in itself is not simple, but quite complex. We call this almost like a Christmas tree. It looks like that. And this has to be an exquisite arrangement for it to function properly. And in fact, in mouse models, what we can show very nicely with this kind of a technology is how the cells are shrinking away, and those Christmas trees are not looking quite as decorative as they should, and they are losing the connections. And it's telling us a lot about how early that is happening, at least in our model systems, and how we can repair that. One of the drug contracts that I know the Philadelphia chapter is supporting, that of Steve Burden, is actually looking exactly at this problem. He has a very interesting molecule that he thinks, together with the company, he can develop to improve this particular connection. So very exciting that our knowledge has expanded much beyond that motor neuron. It's a dangerous thing because all the systems are all in one thing, so I'm trying to point and continue the slides. But everyone says, well, what causes ALS? And certainly what we do know is that genetics causes ALS, and 5 to 10% of the disease is familial. However, genetics probably also plays a significant part in this 90% of sporadic. It's just a little bit more complicated. In the familial cases, it's more likely just one gene that is a dominant factor for it going down into the family. In sporadic, it's probably a combination of multiple genes. And how the environment fits into it, well, that's an even trickier picture. We know smoking is certainly an increasing risk factor. I didn't even put the list of question marks here. The only definitive one really is smoking. The others are still rather unproven and unknown. Age is certainly also a significant risk factor. And just if you imagine that your body goes anyway with aging through wear and tear, add on top of that a genetic factor that really will then push you to the edge of having the disease rather than not. This is the exciting part of science and drug discovery, and partly due to technologies and partly due to the increased number of researchers. But this is what we had in 1993 when I started in the field of ALS, the SOD1 mutations. We know that it accounts for about 2% of ALS. This number is a little variable. There are some areas where there's much more prevalence of this SOD mutation, especially in the Finnish population. And in some areas there's much less representation. In fact, in the Dutch population there are hardly any SOD1 mutations. However, moving along, and in fact I superimposed them all in this slide for the essence of time, but really in 2003-04 we started to get hints of new genes, almost 10 years later, And now in the last year, we've had this cluster of new genes. So there's just been an exponential increase in our knowledge and clues for how we might think about studying and treating the disease. And this is represented slightly differently. This is looking just at familial ALS. And in the pie chart, we see that although we first had a fairly reasonable chunk, the SOD1 mutation, and my proportions might not be so good actually, the C9 mutation, which was found in 2011, is a very significant mutation, about, in some populations, 40% of familial ALS. Probably on average in the US, around 30% of ALS in the familial cases. The two other genes, FUS and TDP43, each individually contribute about 4%. And then this chunk is still those unknown genes. But my geneticist colleagues assure me that in the few years to come, we will probably know all the genes linked to familial ALS. The technology has moved so fast, we can do whole sequencing so quickly. The likelihood is that there will be multiple genes that affect very small numbers of families. I doubt whether we'll see another mutation that looks so prominent as this one. What is interesting is that this mutation is also found in sporadic cases of ALS, and in the Q&A we can discuss what that means a little bit further. So these are the mutations, TDP43 and FUS, that were actually found very soon after each other and have very complicated biology. And in fact, they work on the RNA processing, a new area of research in the field. And what is interesting, you can really superimpose the two. Their functions are quite similar and yet distinct. Again, FUS and RNA, FUS and TDP43 are both found in the familial cases, but also actually in sporadic ALS. And what was interesting, and in fact how they were both found so quickly, is that they are structurally similar. All you have to understand from this is that the red domain is the same in both, and there are similar domains, and they were actually found because first TDP43 was found, and then they thought, let's use that structure and see if there is another similar gene that might be involved in ALS. And now I realize I've got to go here to show you the movie, but why I show this is that really, because RNA processing is an incredibly complicated biology, and basically it's happening in every single cell of our body as we sit here all the time. And so what these things are doing, the C9, the new gene I'll tell you about, TDP43 and FUS, still remains slightly unclear. But as you see things moving in, these proteins have to bind exquisitely with each other for them to do the right function. This is called a process of transcription. It's taking messages from our genetic information and neatly packaging it so that we then get to the further process, and I'll quickly switch on to the next process. And this is a process, also RNA biology, called translation. And now at this point, you've got the messages from the genetic material, and we have to make the right structural proteins in the body. And so all it is to say is that there are many interactions that have to happen exactly correctly for us to transcribe the right proteins. What TDP43 and FUS are really doing when they have mutations, we don't understand. But this is all the biology that we're trying to figure out. Who are these mutant proteins binding with? Is it because they now no longer function properly, which we call a loss of function? Or have they got something new that they're doing something different? So it's such a complicated biology, it's easier to visualize. Stop that. Actually, I think if I just go forward. There we go. Added to that complication, and we talked really about the motor neuron system, the motor biology. Actually, what we also realize more and more, and because of genetics, that this is also a disease that can combine with frontal temporal dementia, and many of you are very familiar with that now and may have heard about that. All these things here, tau, FUS, TDP43, are interestingly found both in abnormally arranged, both in frontal temporal dementia and in ALS. And what we recognize now, that it's more a spectrum of disease. You may have the pure ALS on the one side, pure frontal temporal on the other, and a combination of the two. And that's really why the C9 mutation was so interesting and took so many years to identify. Because in these very, very large families, what sometimes happened is that in one generation you would see ALS, the next generation you would see FTD. And because frontotemporal dementia, FTD, was not so well recognized, it almost seemed that that case wasn't perhaps a familial case. Finally, when it was all pieced together, besides other challenges, we could figure out that this was going to be an incredibly important gene, and I'll touch again on it a bit later. So here are the model systems that have been developed because of genetics. And one might wonder, why so many different and what is the value of them all? Well, firstly, this is here a motor neuron in a dish, so it's very valuable to try and test things in quick format in culture dishes, but of course it's so far away from the human system that it's somewhat artificial and we have to relate it further. We have rat models and mouse models. We have worms, the C. elegans, and they have a very defined motor neuron system that you can see in live time. And in fact, when you make a mutation in these worms, the SOD1 mutation, they can't move as well. So in some ways, they mimic a phenotype that is important to us. And we then can, in a dish, put drugs on and see which drugs change that phenotype. So they've been very valuable. You can see here how this is a living worm, but you can image it in live time and see its network and the nervous system and where the SOD is expressed. We also are working on flies and have learned some very interesting things from those models. So why is it so difficult to develop treatments for ALS? One, it's a complex biology, and we're only just beginning to understand it. The other is that the drug discovery process is actually an incredibly difficult thing too. If we think of Alzheimer's disease and the billions that have been spent in the industry, and really the lack of treatments there, it's a very tough task. Drug discovery is all about first finding a target, and here we could, for example, say SOD1 is an example of a target. It then needs to go through some kind of a design of a drug, something that interacts with that target, so chemistry has to go on around that. Once we have a chemical compound, it then needs to be tested in model systems. It needs to be tested for safety before it can go into clinical trials for FDA approval, and then it goes into patients for observation. And as we know, and with what happened with the Biogen trial, that trial failed after several years, very rapid enrollment, very effective study, but it wasn't successful. And so we have to think carefully why and take it back into the drawing board, either to improve drugs around that pathway or to think of new pathways. So it's really a constant process of trying to improve and figure out why it is that a study like that has failed. I think the encouraging news for us is that although we might have thought, because ALS is not a blockbuster disease in the sense that, like Alzheimer's, there are many, many more people living with the disease, there are many, many companies, and this is not even a complete list, who are spending significant resources and very interested in working in the ALS disease space. So this is very different to several years ago when only a few companies were focused. Bristol Myers Squibb does not have a program at the moment in ALS, but is very, very interested and talking to us about the possibilities. Pfizer is developing has a rare diseases unit and is developing programs and thinking about ALS. So there are many companies, both small and large, that are very invested, and often we don't fund them. We do fund some small biotech companies, but they partner with our academic colleagues and will provide service in kind. So we'll do many of the studies at no cost to us. But what we're left with over all the years of really intensive research is quite a challenging picture. And I only put this up here because this shows you the many components involved in the disease, the motor neuron, the astrocyte, the microglial we'll learn about a lot more just now, the muscles. And the question is, which is the best target for us to develop therapies around? Reminding you that the target is the starting point for drug development. Which one is the important one? If we think of dexpramipexole, which works at the mitochondria, very important powerhouse energetic cells that we need, we know that they are compromised in ALS by doing measurements for mitochondrial function in patients. We've seen them visually that they go wrong in the SOD mouse model. Why did the drug not work? Did it not work because it isn't an important mechanism at the time that we treat? So maybe later in the disease, we can't repair that mechanism, we can't improve the mitochondria sufficiently to overcome the dying motor neurons at the stage that we treat? Is it because the drug just didn't get there sufficiently well? We have a lot of questions, unfortunately, at the end of a trial, and that's something we really want to try and change. And what do we need to help us along that way? And I think it's biomarkers. Biomarkers are signatures of either something in the blood, something in the fluid around the neurons called the cerebral spinal fluid, either brain images, but something to tell us that a person with the disease has a different profile to a person without the disease. If you treat with a drug, that person's profile changes favorably so. And I mentioned neurofilaments to you earlier. These are very important structural components of cells. And in fact, what we found in the mouse model, work by David Warshock and Jerry Shaw, that over time, especially at the ages, at 90 days, these mouse models have hind limbs that are dragging. So when they show signs of weakness, the neurofilament levels, this in particular is one subunit of it, start to increase significantly. What that might just represent is that because cells are dying, we are getting more of this particular protein. But that could be an important finding, but not very useful if it's only happening in a mouse model. So we looked further than that, and in fact see in ALS patients that compared to control subjects, and this is over time between 1 to 4 months, we see variability, admittedly, not the same in every patient, but quite a lot higher levels of neurofilament. Once it's high, it seems to stay high, unlike in the mouse model where we saw this curve going up. Over this, at least this period of time, because we don't go in that early as we do in the mouse model, we see a fairly much a plateau. And even more interestingly, and recently published by Kevin Boylan, it's appeared in a very small subset, so we have published it, it's public knowledge, but we don't absolutely feel confident until we've done a bigger study, that if the neurofilament levels are high, it usually follows that the prognosis is short, so the disease is quite rapid. And these are important things to know, especially when we learn more from Dr. Berry about trials and trial design, and the fact that although ALS is one disease, it's probably a spectrum, with some slow progressing, some fast progressing, some bulbous, some limb, and these kinds of clues might help us actually in clinical trial design and building subsets of patients that we might think about treating. Imaging techniques are really starting to improve significantly, and researchers like Dr. Benneter are also developing imaging techniques that have always usually been in the brain, but now for the spinal cord, because we obviously know that the motor neurons are being lost in the spinal cord. We match physical changes with chemical changes, and he's actually doing this now in a large cohort of patients who have not yet shown disease but carry the SOD1 gene, to see if we can find very early clues before someone has any physical signs. This is work by Martin Turner at Oxford, who's doing fabulous work with different brain imaging in his team. And it just is to illustrate here that you can also do all kinds of live imaging of the brain. And previously, in the past, we could only make these judgments right at the end of disease and show, in fact, he shows here that where the damage is, we can now show that in a living person at a much earlier phase. And so we can use these images here to tell us something about the progression of the disease and hopefully eventually find treatments. I will remind that in the field of multiple sclerosis, which itself also was a very untreatable disease not so long ago, it made huge advancements because of these kind of imaging techniques, where they could finally see significant changes when they did these brain images when treating with a compound. And so not having to wait for very long trials, but in a shorter period, being able to say whether something is working or not. So Biogen actually was a company very focused on multiple sclerosis and has shifted or has added to their portfolio ALS. And I think it's tremendous, even though the trial has failed, that they are still very committed as a company and are determined to find treatments for ALS. This is again another imaging technique that is being developed. This is in a rat model, and I highlight this because this will be an outstanding biomarker for us to understand how glutamate is engaged and involved in ALS. And this is a ligand that has been developed, a technology that's being developed around a transporter for glutamate that sits on the astrocytes. And the only point that I really wanted to raise with this is that not so long ago, we also had the disappointing news that the ceftriaxone trial had failed. And ceftriaxone, some of you may have known, came out of work from the NIH and ALSA, where we did these screens and found it a very interesting, normally an antibiotic, but because of its structure had other functions, that this might be useful on the transporter. But again, in the study, we didn't really have a biomarker, so we were left with the feeling, was this pathway, is glutamate toxicity an important pathway in the disease? Did the drug get to where it should have in the right amounts? And did we actually affect the important pathway? Had we got this ready in time, and it's only just going into safety testing in patients, because once you develop this for an animal model, you have to be sure it's safe in humans. Had this been ready, we might have been able to understand better what had gone on in the trial. So the goal for our programs moving forward, as much as we can, working with experts worldwide, is to build the biomarkers, if possible, at the same time as thinking about how we will build a clinical trial. And not always easy. This took at least 8 years to develop. So some of these things are very difficult technologies. We are also in a field where the disease being challenging, we won't stay with just one idea of, we'd love to get a drug that you can take like an aspirin once, 3 times a day. But on the other hand, we can't rely on that being the route that is going to be successful. So we've done very innovative, we, the royal we, the community, have built some amazing tools to try and treat in a much more creative way. And this is what I had mentioned earlier, the antisense oligonucleotides. These are an approach where you get a message. I'll show you, it's complicated to explain without a picture. I'll tell you that just now. Stem cell therapy being another area, and gene therapy. I will talk a little bit more about stem cells in a moment. So this is what I needed as my crutch to try and explain. So if you remember that you have obviously a long string of messages that is then going to translate into this is SOD1. And what you can do with antisense technology is you can make a little piece of message. That message then binds to the message that is required to make SOD1 protein. Once it's bound together, there's a special enzyme that chops it all up and gets rid of it. Simply said, what this technology can do is it can block the production of something you don't want to have. And in this case, we don't want to have SOD1, mutant SOD1. We had proven, and I had done this work together with Don and others, that if you get rid of SOD1 in a mouse model, the mouse model does not get ALS, not get a robust disease, so we can get rid of at least some of it, because this antisense technology will not get rid of all of your SOD protein, but it will dampen it down significantly. So we think very much so that this is a safe technique. I belabor that point because I know that we now also mentioned the FUS and the TDP43 genes, and unfortunately with those two, I don't think we can go and do this. In fact, I know we can't do this, because if you knock it out of a mouse, the mouse is lethal, because it's so important. I showed you that image. It's so exquisitely involved in a fundamental biology of the cell that we can't knock it out. We have to do something different in those cases. But in this case, we certainly think it's very promising. And this was the first disease that this company, ISIS, launched in for a neurological disorder. The drugs that they had marketed before were for ophthalmology, for eye diseases, because the eye has a very focal region. They were able to do it for one very specific region in a safe way. Of course, this is a much more complicated technology, and it wasn't so clear that if you put this kind of thing into a person, that it actually, remarkably, when you put it into the spinal fluid, it goes all over the body. That was something we wouldn't have guessed, really, before doing it in a rat model. And this is showing you exactly that. It's showing you that what is done in the clinical trial is that through just and this is, I'm not the clinician, but I believe it's very simple, and you really just put it through into the spinal fluid area here, and it gets to all over to the motor neurons, and that's where it next has to act. Quite remarkable technology, actually. And this went into clinical trials and was published. So why has it not gone further than that? And I know many, and I know that there have been from the chapter significant contributions financially to the study. Very, very pivotal for the field in general. It's now a trial for spinal muscular atrophy, a childhood motor neuron disorder, thanks to the work that was done here. It took 5 years to get from bench to bedside, which actually in the scale of drug discovery is incredibly fast. That is phenomenal. But in those 5 years, and with now attacking diseases like Huntington's, SMA, myotonic dystrophy, we have advanced again even further in the technologies, and the company has been able to improve the chemistries in such a way that they feel they can do it better. Remember that once you are going in for this treatment, and if it's effective, we don't know that yet. The trial said it's safe. We don't know if it had any effect because that wasn't the goal of the trial. If we're going to be doing this, it will be long-term treatment. And so for a short period of time, we could probably tolerate some side effects, but for a long period of time, we want to minimize side effects. They have chemistries that are much better now that will improve that, they believe, and felt it was more prudent to go back and build better therapeutic molecules before going into very expensive phase 3 trials where you probably wouldn't go back anymore. And so the timing is good. We are confident that they are progressing quite quickly, and we hope to be in trials within the next year or so, and I will keep you posted. I don't have too much time to tell you more about this, but really just to say that this was an incredibly exciting finding. This here is in a Finnish population, and suddenly from doing an international consortium that really Ellen mentioned I was involved in building together, we found this enormous peak, and this was a C9 mutation, which we now know is very, very prevalent. It took us several years, 3 years after that, to still really work hard at trying to identify it. And partly these are the 2 publications that came out from Bruijn Trainer and Rosa Rademakers. Partly the challenge was a new technology, but all I want you to go home with is to know that this is where the problem is. This is before the area where, so normally what you do is you get the production of your protein. We've talked a lot about how the messages are going to be made from your DNA. At this point, it starts and makes protein. So this abnormal change is in a region before it starts to make protein. So what we have is suddenly an incredibly large expansion. Instead of being less than 20, it's thousands long in an area before the protein is made. And we think that it's involved in making clumps of RNA that is actually basically not wanted and causing problems in that cellular system where I showed you that everything has to interconnect and move in the right way to get your building blocks of the body. And what is most important message here is that we're not waiting to, we are doing the work, but we are not going to wait to understand all of this. ISIS has now developed the antisense to try and block this little piece here. Well, it's actually very long now in the mutant disease, and we hope to use the same antisense technology to treat the C9 patients. It's still early days, but it's moving a lot faster than what we have ever known in the past, so we're very hopeful about that. I think I'll leave this more or less to questions, only to highlight that we obviously are very excited that the NeuralStem trial is going into the next phase. More people will be enrolled to see if there is any effect and benefit. We so far know it's safe, this very invasive technology. And Klaus Svensson is building his tools and is about a year away from the clinic to use a slightly different technology. He's going to use this as a vehicle to introduce a trophic factor, GDNF, which is very good for motor neurons, and we hope will increase their survival. And that is planned for 2012, late 2014. He's been having discussions with the FDA. And I'm going to really move along with this, only to emphasize that we are excited to support the NEALS network and really help build clinical trial sites throughout the US that can very quickly and rapidly enroll with the right standardization and good results for the analysis. We hold monthly webinars. I know many of you here participate. We're excited to be able to bring the scientists and clinicians to you and have Q&A and discussion, so always welcome you. If you've missed them, you're very interested in them, they're on our website, they're recordings and then summaries to make it a little easier to follow. We also have a clinical trials information site, and Devin Punch is a tremendous investigator who helps you, guide you, suggests which clinical trials might be of interest to you, because this is also a very complicated arena.